Hello guys, welcome back to my channel BioFreak. In today's lecture video, I'll be talking about replication in eukaryotes, that is in yeast or Saccharomyces cerevisiae. Now in Saccharomyces cerevisiae, we have ori that is called as autonomously replicating sequence. Now this autonomously replicating sequence has three domains, that is A, B and C. Now A is ACS, that is ARS consensus sequence. ARS is autonomously replicating sequence. Consensus sequence is A domain. B domain has B1, B2 and B3, and there is C domain. Now A and B1 of B domain together form origin recognition sequence to which origin recognition complex will go and bind. Origin recognition complex is basically made up of 6 polypeptides of CDC1 to 6. CDC is cell division cycle proteins there. Okay. Now once they go and bind to origin recognition sequence, mini chromosome maintenance protein, that is MCM2 to 7 or helicase in eukaryotes, goes and binds to that particular place, okay, of B domain. Now this also involves addition of CDT1. CDT1 is chromatin licensing and DNA replication factor 1. And as I mentioned before, that there is origin recognition complex made up of 6 polypeptides. It has CDC6 as well, right? So it also has ATPase activity. Now hold together, hold this complex plus your helicase and CDT1 together, they form pre-replicative complex. Now, in order to activate the complex, you require cyclin dependent kinase 2 and DBF4 dependent kinase. And helicase is activated by genes and CDC45. Okay, genes is basically GINS and CDC45. Now you enter into the S phase. Whatever happened before this was G1 phase. Now you enter S phase of cell cycle. Now what happens is helicase will start unwinding the DNA. As it unwinds the DNA, you will get single stranded DNA, right? So this single strand of DNA has to be stabilized by DNA binding protein, single strand DNA binding protein. It is done by RPA or replication protein A. Now there'll be two strand, one will be leading strand, okay, wherein there'll be 5' to 3' polymerization. So it is done by delta polymerase delta. Now previously it was thought that polymerase epsilon carries out polymerization of leading strand, but recent studies show that polymerase delta carries out polymerization of both the strands, lagging and leading strand. To increase the processivity, genes, okay, GINS, although I have written it as gene, okay, it's GINS, it increases the processivity of this polymerase. Now talking about the lagging strand, as we all know, you require primers there. So these primers are added by primase, which is pol alpha or polymerase alpha. Polymerase alpha has primase activity as well as polymerase activity. In primase activity, it will add primers, okay, and these are also known as initiator RNA, and later it will add initiator DNA, it will leave. Now polymerase delta will carry out polymerase activity. It will start synthesizing the DNA. It will complete one by one Okazaki fragments, okay, and there should be something called as, sliding clamp and clamp loader, right? So here uh, RFC, okay, RFC, that is also known as replication factor C, will load PCNA. Okay, now PCNA is proliferating cell nuclear antigen. PCNA is sliding clamp and clamp loader is RFC. Okay, now talking about the removal of primers, once you complete the polymerization of lagging strand, there will be still primers which are to be removed. That is done by flap endonuclease 1 in eukaryotes. Later, Pol delta will fill the gaps again, and DNA ligase will ligate the last phosphodiester bond will be formed, and it will ligate that fragment. So that's how the replication takes place in eukaryotes, to summarize in brief. Okay, I hope that flowchart was helpful, and you guys can draw it on your own and summarize it and understand the concept. Thank you.